All right, welcome. Today, we're going to be construction workers, but on a microscopic level. We're building a cell's first line of defense, its gatekeeper, and its main communication hub. But listen, this isn't just some static brick wall. We're talking about the cell membrane, a structure that's way more like a living, thinking city border than just a simple barrier.
So let's kick things off with a really basic question. Just think about it for a second. A cell is mostly water, and it's full of all this intricate machinery. And the whole thing is floating around in a watery environment. So what actually gives it shape? What's holding all that amazing complexity together and preventing it from just dissolving into a tiny puddle?
And the answer, as you've probably guessed, is the cell membrane. But I want you to ditch any image you have of a simple plastic bag. This barrier is incredibly smart. It's flexible, so it can bend and move without breaking, and it's interactive, constantly talking to the world around it. Okay, let's get into it and see how this incredible wall is actually built.
So, any wall needs bricks, right? But in the world of the cell, these bricks aren't hard and rigid. Nope, they're kind of squishy, they're always moving, and best of all, they build themselves. We're talking about the absolute foundation of the membrane, the lipids.
Now, the most common type of lipid here, the phospholipid, has what you might call a split personality. It's got a hydrophilic head, which absolutely loves water, but then it has these two hydrophobic tails that can't stand water. They will do anything to get away from it. And this simple love-hate relationship with water, well, that's the secret to the entire structure.
This is where the magic happens, self-assembly. The second you drop these phospholipids into water, they just spontaneously arrange themselves into this perfect two-layered sheet. All the water-loving heads automatically face outward, towards the water inside the cell and the water outside. Meanwhile, all the water-hating tails get tucked safely away in the middle. And boom, just like that, you have an instant waterproof wall.
But you know, it's not a one-size-fits-all kind of wall. The membrane uses different types of lipid bricks for different jobs. You've got your standard phosphoglycerolipids keeping things nice and fluid, but then you have tougher ones like sphingolipids. These guys like to cluster together to form these little floating platforms or rafts where proteins can hang out and send signals. So this diversity is super important. And those bricks with the little sugary hats on them, the glycolipids, yeah, we're going to circle back to those in just a minute.
Okay, so our wall is fluid, but we can't have it being too fluid or too rigid. It needs a manager to keep things at that perfect consistency. And this is where the unsung hero of the cell membrane comes in, cholesterol.
I know. When you hear the word cholesterol, you're probably thinking it's a bad thing. But inside the cell membrane, it is an absolute superstar. At warm temperatures, like inside our bodies, all those lipids can get a bit too excited, moving around way too freely. Cholesterol just wedges itself in between them, slows them down a bit, and stops the membrane from turning into a leaky, disorganized mess.
But here's where it gets really cool. It does the complete opposite when things get cold. When the temperature drops, those lipid tails want to huddle together and freeze solid, which would make the membrane shatter. So cholesterol gets in the way again, but this time it acts like a little spacer, physically preventing the lipids from packing too tightly. It keeps the membrane fluid and flexible, even when it's cold. So you see, cholesterol is like the perfect thermostat for the membrane. It keeps the wall from ever getting too floppy or too stiff. It's always just right. And this is totally different from the health issues of high cholesterol in our blood. At the cellular level, life would be impossible without it.
All right, so we've built the basic wall, and we've made it stable. Now for the fun part, decorating the outside. But these aren't just for looks. These decorations act as the cell's ID system, its protective fuzzy coat, and even its welcome mat. Let's talk about these sugary ID badges.
This entire fuzzy outer layer of sugar chains has a fantastic name, the glycocalyx. It literally just means sugary coat. These chains, called glycans, are attached to the lipids and proteins that are sticking out of the membrane, creating this unique fuzzy surface for every single cell. So what's the point of this sugary coat? Well, it has 4 incredibly important jobs. First up is recognition. It's how your immune system can tell a friendly cell from an invading bacterium. It also offers protection, acting like a physical bumper for the cell. It's also vital for adhesion, which is how cells stick together to form tissues. And finally, it's all about communication, acting like an antenna to pick up signals from the outside world.
And if you want a perfect, mind-blowing example of this in your own body, just think about your blood type. Whether you're A, B, AB, or O is decided by nothing more than the specific type of sugar badge that's displayed on the surface of your red blood cells. That's it.
It's a fantastic illustration of how cells use these sugars for identification. Okay, let's take a step back. We've got our squishy, self-assembling bricks, we've got our temperature manager, we've got our sugary ID badges. Now, let's zoom out and put all these pieces together to see the final masterpiece.
This whole concept, this beautiful, complex, moving structure, it has an official name. It's called the fluid mosaic model. Fluid because all the lipids and proteins are constantly moving and shifting around, kind of like boats floating on a sea. And mosaic because it's made up of all these different pieces, the lipids, cholesterol, proteins, and glycans, all fitted together to make one functional whole.
And that right there is the big takeaway. The fluid mosaic model just perfectly explains this dual personality of the membrane. It's strong and stable enough to hold the entire cell together, but at the same time, it's incredibly dynamic, always changing, always interacting with its environment. It's really the perfect solution to that problem we started with.
So, I'll leave you with this final thought. We've seen that all our cells are covered in these unique sugar ID badges for recognition. So, what do you think happens when those badges get damaged? Or what about when a cancer cell starts making weird new badges to try and disguise itself? The implications are absolutely huge, affecting everything from immune diseases to how we detect cancer. Definitely something to think about.